So continue our discussion on microbial growth. Let's take a look at generation time. So the good book gives a general guideline. The time required for a cell to divide is somewhere between 20 minutes to 24 hours. Pretty broad guideline. Under ideal conditions can be even faster, and under not so ideal conditions can be much longer than 24 hours. But good place to start here. So binary fission, the mechanism that bacterial cells use to divide, doubles the number of cells with each new generation. So that means we have a logarithmic growth curve. And if you wanted to calculate the number of cells, then you would have to know how long each generation time is, and then take 2 as your base, and then the exponent will be the number of generations, so 2 to the number of generations, whatever that number is. And if you plot something like this out in biological systems, if cells divide and they grow by 2 to the whatever number of generations, then you have a typical J-shaped curve. So it would look like this. Typical growth curve goes up like what we see right now with the coronavirus cases. Because you're going over so many orders of magnitude when you're following a growth curve, a lot of times these growth curves are represented logarithmically. So if you do that, then now if you have a straight up J-shaped curve, a logarithmic curve, then you can make your X and Y axes, you can add the logarithm here, and then you will flatten out or linearize that scale. Will look like this. So the J-shaped curve, this is a linear representation of the growth of the bacteria, and here this would be a log plot of the bacterial growth. So this slide shows you a visual representation of the numbers. You start out with 1 cell, and the generation time is 0, so it's 2 to the 0. And there's your 1 guy right there. And then you double, and that would be now 2 cells. And so now we have 1 generation, so now we have 2 to the 1 power. So we have 2 cells. And then we have, with the next generation, 4 cells, so it would be 2 squared. And there's all 4 guys. And then 8, 16, 32. And here you see the logarithm, 2 to the 3rd, 4th, 5th, and then you can see the numbers of cells as they go up exponentially. So here will be more numbers for you. Generation numbers now in steps of 5. Here's single ones between 15 and 20, but you can see how all of this follows. It's true as a base, and then the exponent is the number of generations. And then here is the number of cells represented, and here the log to the base 10 of the number of cells. And so if you then take that again into a plot, then you would see the J-shaped curve right here. So J-shaped, that means you have a linear x-axis right here. And if you're representing it with a log, then you're linearizing the plot. If you're interested, you can take a look at the World-O-Meter accounting of the coronavirus cases and case numbers and deaths. And then you can choose on that website or on the app, you can choose how you want to see the data, and they will ask you, do you want a linear representation, which then gives you this J-shaped curve of the cases, or you want to see a log plot, and that will give you then a linear increase of the numbers. Next, we're going to take a look at the phases of growth in a bacterial culture. It always goes through the following 4 stages. We have a lag phase, a log phase, stationary phase, and a death phase. The lag phase is when this culture starts out, first with 1 bacterium, then 2, then 4, then 8, and 16, 32. You really don't see much. If you go back here to this, take a look. This whole time right here, you really don't see much growth at all. No turbidity. It looks like as if nothing's going on. That's the lag phase. And then we're going into the log phase. That's when we're bending and we're going up exponentially right here. That's the log phase. And then the stationary phase is when the culture starts running out of space and food, and that's followed then by a death phase where you have more cells dying than new ones being manufactured. So here's the typical stages shown, starting out with lag phase, then log phase, then stationary, and then death phase. So always going through those 4 stages. Please make sure that you know these. There will be some questions on the next exam. So this is not going to be part, of course, of our Unit 1 exam, but this is definitely information to know for the next exam. So here's something that is also part of this week's lab, the direct measurement of microbial growth. And hopefully you got a chance to take a look at the lab that I posted for you. That was making serial dilutions and figuring out how many cells do we actually have in a culture. There are several methods to do that. If you want to count microbial cells, not thinking about any particles that are just in the test tube that cause maybe turbidity, or dead cells that no longer are able to produce a colony. So if you just want to go after live microbial cells, then the best method is to do a plate count. And there you do a serial dilution, you're plating out the bacteria, and then you count the colonies that you're getting, and then factor in the dilution factor, and you get how many colony forming units you have in your test tube or in your flask. Filtration is another method. There you take a certain amount of fluid where you're suspecting there's some bacteria in there. You filter the fluid amount through, and then whatever colonies get stuck on the filter membrane, you will put the filter membrane on some agar and then watch how many colonies you're getting. That's especially good if you are expecting low numbers. Let's say the water district wants to test whether there are any bacteria in the water. You would filter, let's say, a liter of water and see if there's any bacterial growth on your filter afterwards. The water district might also do something called the most probable number method, the MPN method, and I'll show you that on a different slide. It's kind of hard to explain without having a diagram. Or you could do direct microscopic counting, which is kind of tedious, especially if you have, it's just really hard because they're moving and it's hard to count them when they're live. So let's start with the plate count. So you need to do a serial dilution in order to have plates where you can actually count colonies. If you're just taking a straight up culture and you're plating that out, you're going to end up with a lawn of bacteria, and there's no way you're going to count single colonies. So you need to do a serial dilution and then put a certain known amount of liquid from the diluted medium onto an agar plate, and then you're going to have to wait like 24, 48 hours, and then you can count the colonies that have been manufactured. There are 2 ways you can do this. You can either do a pour plate or spread the bacteria on the surface. The pour plate means that you have sort of a thin layer of agar in your plate, then you put your bacteria into the mix it with the agar and pour the rest on top, and then you have the colonies growing in the medium and on top. It's probably better to do the spread plate method because some bacteria, they really don't like to grow inside of the agar medium. So here is an example for the spread plate method. You're doing your serial dilution, so you have an original broth, some original culture, and you're taking 1 milliliter of that and put it into 9 milliliters of water or some phosphate saline, and then you're plating 1 milliliter of that. Most likely it's so many bacteria that you don't even see single colonies. It's just going to be a lawn of bacteria, like you can see here, so no way to count that. Then here you have your next dilution would be a 1 to 100 dilution. So you're taking 1 milliliter of this stuff and put it into this test tube, which also contains 9 milliliter of broth or some buffer. And now you have 10 milliliters of 1 to 100 diluted material. Take 1 milliliter of that and put it on top of some agar, spread it out, and see what happens. Most likely it was still too many bacteria to count. You do this again. Now 1 milliliter of this, and you put it into 9 milliliters of broth. You get now a 1 to 1000 dilution, and that may give you some colonies that you can count. And you do this 2 more times with every dilution factor. If you dilute by 10-fold every time, then your number of bacteria, they should go down 10-fold. In other words, if you have 3 guys growing right here, so then in a 10-fold more concentrated solution, you would expect about 30 colonies, and you would expect about 300, so approximately. And these are just too thick to count at all. Yeah, that was the experiment you were supposed to do this week, but well, it was the virtual lab, so I hope you got a chance to take a look at that. Here are the 2 methods shown. The much more common method is to spread the bacteria on top of the agar. So that means right here, so you would take from your diluted test tube, you take a certain known amount of the diluted bacteria, you put it on your agar surface, and then you use a spreader to spread the bacterial solution, and then you should be getting isolated colonies. Another method would be that you are putting you're mixing basically the bacteria with liquefied agar, and then you're sort of spreading it with the liquefied agar and let the bacteria grow. They will grow in the agar and on top, so you can see that right down here. It grows. Some colonies will grow in the depth of the agar, and some will just grow on top. Next method is the filtration method, and there it's usually for larger amounts of fluids where you want to find out if there are any bacteria in there. And here's what that could look like. So you're filtering the bacteria, and then the bacteria they're stuck on that membrane that you use to filter the stuff through, and you're putting your membrane on top of some agar surface, and there you have your colonies. In the most probable number method, that requires multiple test tubes. You count the positive tubes, and then you compare that with a statistical table. And let me show you what that looks like. So here you have always sets of 5 test tubes that you inoculate, and then you count the number of positives. So here is some growth medium that changes color when you have bacterial growth, and so the number of yellow test tubes will be counted. And so here you have your dilutions. And then here are your 5 test tubes. You can see with the set 1, every single one of these test tubes shows growth. In the second set, you have 3 out of 5 test tubes show growth, and in the set 3, you have 1 out of the 5 test tubes shows growth. You have to take then the result from the experiment and compare that to your MPN table. And so we are here, we have 531 was our result from the 3 times 5 test tube sets, and that is an MPN index of 110, which means that with 95% confidence we can say that per 100 milliliters of our solution we have 34 to 250 colony forming units. So in the direct microscopic count, you take a volume of a bacterial suspension, place it on a slide, and then calculate the average number of bacteria per viewing field with this formula. It's not something that I was going to do in the lab. So here will be the direct counting, direct microscopic counting. It's kind of hard to do because these bacteria a lot of times are moving, and plus you don't have much contrast when you're counting live bacteria. Now here, estimating bacterial numbers by indirect methods, that would be the turbidity measurement using a spectrophotometer, and that was also part of the lab that we were going to do this week online. And there, keep in mind with the turbidity measurement, you are not measuring live bacteria, you're measuring particles, you're measuring the cloudiness. And so what you're measuring here is how much light is blocked by your culture being there. You can tell that here the light is unobstructed, so you get 100% transmittance, 0% absorbance. Down here you have particles in your solution. You don't know whether they're alive or dead, but nevertheless they will scatter the light, and what you are measuring, the amount of light that's coming through, is much less. And so now you're going to have less than 100% transmittance and some amount of absorbance, and that can be correlated back to bacterial numbers.